So my talk is basically on genome sequencing. Let me ask you this question. Have you wondered, or have you had this question any time in your life? Why are each of us different from others? Of course, we may have some similarities, but at least a few things could be unique for each of us. So why are we unique from others? Did you have this question? Yes. Similarly is the case with crops. Crops also do have unique features. For example, if you take chickpea, this is nothing but the kabuli or the chana which you eat in your chole and bhatura. So in each of these species, we have different varieties where we have unique features for each of these varieties within a species. Why are we different? Now let me ask you one more question. Is mankind more powerful or the nature? Okay, both are powerful. Which is more powerful? Nature, of course. Can you imagine nature has packed all this information of DNA in way back in how many years? If you can see here, the figure, the diagram is not very good. It is packed around 4 billion years ago. 4 billion years ago. Can you imagine where this 4 billion years ago is on the timeline? And then where human evolution began, that is 6 million years ago. So now can you imagine these both timelines, 4 billion years and then 6 million years of human evolution. Let me come to our human evolution. So we took 6 million years and to generate this modern Homo sapiens, or to develop into this modern Homo sapiens, we just took 300,000 years. I'm telling you again, or I repeat, just 300,000 years. Can you imagine now where from we have reached 6 million years and also 300,000 years. So the word Homo sapiens comes from a Latin word. Homo is human and sapien is wise. Wise human. That's how we named it, Homo sapiens. Now let me tell you the story of DNA. So first, the structure of DNA has been identified by Rosalind Franklin in 1950. It is very tedious process at that point of time, where we didn't have any technologies with us, where we didn't have any photographies or so. So she just did it with X-ray diffraction image and identified that DNA has double helix model. And then Dr. Craig Venter, he decoded the DNA into 4 simple letters, that is A, C, G and T. So it happened in 1990. Can you imagine that it took almost 40 years to come from 1950, where the structure of DNA has been released, and then to decode the DNA, it took 40 years. And then let me tell you again that before we have all these technological interventions, to build a human, it takes 3.2 billion letters. A whole human being has 3.2 billion letters. That means you have 3.22 ACGT code within your body. And then we don't have data analytics or big data analytics at that point of time, or any bioinformatics pipeline to decode all this information into genes and so on and so forth. So they printed all this 3.2 billion letters in almost 262,000 pages. You can see the books here in this image. How many books were there? Almost 22 books were printed with all this 262,000 pages. So Craig Venter has ventured on this, and he contacted different people around the globe, and they began to start the Human Genome Project in 1990. A consortium of 20 international organizations have come together to decode this human genome. It was a very big collaboration at that point of time. 20 international research centers have come together. However, while beginning in 1990, they didn't have any particular plan. So they just began to decode this genome, human genome. And then it took almost 13 years to come up to the stage of decoding entire human genome. In 2001, almost about 90% of the human genome was published. This was the first draft. Of course, there are few gaps within them, and these were filled by different technological interventions, and 100% was released in 2003. It took 13 years, and can you imagine the funding? It was almost $3 billion. So that means for every letter it took almost $1 to decode what is there within the human genome. And they have identified 30,000 to 50,000 genes which were responsible for different traits within human being, like the hair color, eye, skin color, so on and so forth. This was published in 2001 in high impact factor journals like Science and Nature, and then the complete genome, as I was mentioning, was available in 2003. Of course, genomics was a toddler field at that point of time when they began, where they didn't have any plan, but they learned from their lessons and slowly improved on how the genome can be decoded. So the message from this slide is, children or students, if you don't have a plan, that's fine, but just begin and you can meet the success. These are the genes that are called the miracle of life. They have decoded these 30 to 50,000 genes for different traits of human being, and these genes, or the pathway of these genes, are called the miracle of life. So I call it as a valuable gift from your parents. The 3.2 billion letters which are coming from both of your parents is the valuable gift of your life. You can improve it or you can destroy it with your habits or day-to-day life. For example, if you have bad habits like overeating or snacking so much, and then you can deteriorate your health, and if you have an activity or good eating habits and so on, you can improve your health. So always keep yourself healthy, active, and then you can treasure that gift which you have received from your parents. Now coming to different bad habits. I always insist on this slide, the screen time versus the genes. Why? You must be wondering why this lady is talking about screen time and how is it associated with human genome or genome sequencing. Yes, it is associated. So there are several adverse effects like sleep deficiency, reduced bone density, sedentary behavior, obesity of course. From obesity, as you can see here, the DNA gets damaged to some extent and then disturb the cell metabolism within your body, and then it may lead to metabolic disorders, or even it may develop cancers. So children, I am not only focusing on children or students, but also for the adults who are sitting here, please reduce your screen time. Screen time is very well associated with the genes. You must be feeling happy, right, that I am pointing out at elders as well. Yeah, of course, several elders are also now addicted to phone. I take an example of one of my family members. Even while he sleeps, his finger does like this. That's one of the disorder from overscreen time. Now the question comes, can we cure these genetic disorders or diseases by altering the DNA? There is an answer coming from different technologies which are evolving. I will let you know a few examples of genetic diseases. A simple transition from A to T in hemoglobin genes leads to sickle cell anemia. Can you imagine just a single base which alters in your human body would lead to sickle cell anemia in hemoglobin genes? These are called point mutations. Just at one point, if it is altered, your DNA or your code get disturbed and it may lead to different diseases. Not all mutation points are harmful, but of course some are really harmful. Again, here we are talking about A to T base transition, and then when you come to C to T, then it causes rapid aging, progeria. I hope you have known this very well. Children with this rapid aging can live only up to 10 to 15 years, unfortunately. So this all happened with just change in just one base pair. Now we have several technologies developed. You must have heard about CRISPR. So CRISPR gene editing therapies are available. What does this do? So this CRISPR acts as molecular scissors. It cuts the DNA and then these base editing tools can be inserted so that the DNA can be re-returned. So it may not be complete cure, but definitely an improved stage of your health. Okay, we have been talking about bad genes which are causing sickle cell anemia and then the progeria and so on and so forth. Now let us talk about good genes. Did you ever imagine or what genes were responsible for our Ratan Tata, who is chairman of Tata Group? So what are the genes responsible for him to be an inspirational leader, a person who focuses on social impact, and whatever decisions he takes leads to success. So can you imagine what genes are really responsible for these leadership qualities and successful life? There are genes definitely associated and which are identified for the leadership qualities. If you would like to know what genes are really associated within you to prove yourself as a leader, you can sequence your genome. So it took 13 years for the researchers to sequence the whole genome of a human body, but now with the technological interventions, you can do it in just a couple of days, less than 48 hours, with very reasonable amount of price. Now, I would ask a question to you, who is taller, either Abhishek Bachchan or the Big B? Big B? With the name, you can never say he is big or tall. So Abhishek is taller than Amitabh Bachchan. So he is 6 feet 3 inches and Amitabh is 6 feet 2 inches. And about 50 genes were identified from the human genome which are responsible for height or the trait height in human beings. Now, boys must be wondering why always Big B and Amitabh Bachchan, why not actress? So here comes your beautiful actress. Can I ask you who is more fair in complexion, Tamanna or the Kapoor sisters? Whom do you think is more fair? Kapoor sisters, you're wrong. Tamanna is more fair than Kapoor sisters, and about 169 genes were identified for skin color in human beings. Okay, now let's come to what is the utility of this whole genome sequencing in your life. So for example, here I would like to give an example of prenatal whole genome sequencing, where you can identify specific anomalies in fetus. So a pregnant woman, if the genome, prenatal whole genome sequencing is done, a pregnant woman can identify, or the doctors can identify what are the anomalies within the fetus. Then you may decide at initial stage of pregnancy whether this pregnancy can be carried forward or you may restrict it at that particular stage. There are gene solutions available which you can alter the DNA, as I have mentioned earlier. However, we have to undergo this genome sequencing. So there are several other utilities like whole genome for prenatal whole genome sequencing. If you sequence a genome of human being, you can have better health, personalized medicine, etc. for in your day-to-day life if there is any serious concern by the health of the particular individual. Now here comes your favorite food, Hyderabadi biryani. How many of you like it and enjoy it? I see almost all the hands raised. Of course, I am one of them. I really enjoy Hyderabad biryani. And where do you think this flavor of rice or biryani comes from? It is nothing but the basmati rice, which is identified to be flavored, or that flavor comes from basmati. Of course, we add some spices to it to get it more tastier. For example, here what I wanted to say is the rice, with the sequencing of the rice, we identified this OSBADH2. There are several genes which are responsible for the aroma of rice. Similarly, there were genes which were identified for different stresses in plants as well. Like in humans, we have different genes identified for abiotic stresses and nutritional traits in crops as well. Being a crop scientist, I tend to talk more about crops. You must excuse me for that. And here is the whole genome resequencing project of rice. It is an international rice genome sequencing project which identified more than 40 to 50,000 genes. It is one of the cereal crops with very less genome size, just 430 MB. And can you imagine, it took almost 8 years to get all the genome sequenced of that particular small size crop. Of course, wheat is another crop. Now we have several crops already having genome sequencing. Even the minor crops have also been sequenced, including millets. Now coming to gene banks across the globe, like we treasure all the genetic material of humans to utilize that in different therapies, etc. We also need to treasure this seed or varieties which are having genes of our interest, for example, which can combat abiotic stresses and which have high nutritional value. So we have several gene banks across the globe which is conserving around 736 to 10 accessions of cereals, grain, legumes, different tubers, and again root species, tree species, etc. across the globe. Can you imagine 700,000 species were conserved in these gene banks, and they are used for any of our future breeding programs to generate or develop improved or new varieties of these crops. Last but not the least, I would like to stress upon this gentleman, Professor M.S. Swaminathan. Hope you know him. Yeah, he is a father of a green revolution, Indian green revolution, and he passed away recently at the age of 98. But he has left with us a legacy of agriculture research which we can walk upon in coming future. This ends my talk.